So what I'm going to talk about today is some work that I've been doing over the last 5 or so years, trying to understand biodiversity from the perspective not of whole specimen echinoderms, but from the sand they make when they disarticulate upon death. And the reason for this is one of the great driving questions in not only the paleontological circles, but in the biological circles is biodiversity. You can think of this broadly defined as the variability and variety of the life on this planet. Paleo looks at this from a different perspective than a biologist was. We look at large-scale temporal changes in biodiversity, and it is really one of paleobiology's greatest hits. It allows us to understand the temporal shifts in every group of organisms that we have a fossil record for, the recognition of the evolutionary faunas. It's how we characterize mass extinctions and adaptive radiations. Something that a lot of people don't know, some of the real early pioneering work on community-driven database science was done on paleobiology data sets back in the 70s. And that continues through today with things like NMEDA and the Paleobiology Database, which are landmark databases that have been the template for later research. Paleobiodiversity also informs human impact on our current biological crisis. And you could go on and on. It's a very important topic, to say the least. And the summary diagram from Jack Sapkowski in the late 1970s is old, it's dated, but it is iconic. It's one of the most famous figures ever made in paleobiology. And this is nothing more than a histogram. Some measure of diversity along this axis and time here. So things like adaptive radiations go up, extinctions go down, and from that we can categorize, we can quantify major shifts in the Earth's biosphere. Now, really, so if you think about this, all we're seeing is a measure of biodiversity through time on histogram. But that biodiversity pattern that we see is actually overprinted by lots of biases. And biases aren't unique to paleobiology. All of our sciences have biases in them. But bias is something that fundamentally affects how we understand this. And there are 3 fundamental different types of biases that I think you can easily identify in these data sets. The bias that's intrinsic to the organisms themselves. Something about the biology of the organism causes a bias. Biases that are extrinsic to the organism, but are the result of some issues with fossil preservation, the rock record. And then finally, the biases that are associated with the researchers, the study methods, the sampling. So just quickly to look at these, intrinsic biases might be something like the presence of a skeleton. You know, if you look at these 3 organisms, a ctenophore, a comb jelly, doesn't have a skeleton, so its fossil record is automatically in jeopardy. The original mineralogy of the skeleton can be very important. A lot of rocks, like in the Cretaceous, will have both oysters and snails in them. But because oysters are low magnesium calcite and snails are, generally speaking, aragonite, their preservation potential is very, very different. Oysters are beautiful, snails are moldic. The thickness of skeletons, fragile skeletons are subject to destruction, whereas thicker skeletons are not. And the ability to recognize things is often a function of how many skeletal elements they have. Something with a few skeletal elements is good. Something with hundreds to thousands to millions of elements is generally bad. Extrinsic biases in terms of preservation are also a problem. biological and mechanical reworking of skeletons, so organisms tearing things apart or being washed down a stream or ground. Diagenesis and other rock processes can destroy fossils or in some cases enhance the preservation. Odds of burial of organisms. Burial is generally required for preservation, but some organisms and where they live and how they live interacting with the environment are more easily buried than others. Environments that organisms can live in are not evenly preservable within the rock record. Certain types of ecosystems like high mountain slopes are never going to be preserved because they're erosional, whereas places where sediment is being input will have higher preservation potential. And then we have destructive forces on a massive scale, weathering and erosion and tectonics. So these sorts of things and the previous slide are all things that can fundamentally affect our ability to see fossils in the fossil record. But what's nice about these two categories are we understand them. Because we understand them, we can work with them, we can understand how things are missing, and we can mitigate how things are missing using, just simple things like, I wouldn't expect this, so what should I expect if I did see it? Where we're much more blind is the biases that are associated with study methods. So the rock that an organism is preserved in affects whether or not we collect it, because certain types of rocks are impossible to get good fossils out of, so we generally speaking ignore them. Some organisms like dinosaurs, people actually study. Some things like bryozoans, there's 2 or 3 people in the world that look at them. And which one makes up more of the rock volume? Well, it's bryozoans by several orders of magnitude. The way we deal with our taxonomy, whether we overgeneralize organisms, making multiple species into one taxon, or we undergeneralize them, making a single species into numerous species, because of true ignorance of the true nature of things, can fundamentally affect our counts, either overcounting or undercounting. Some fossils are not easy to sample because of other conditions. Most fossils are haphazardly sampled. We simply walk around on our hands and knees, picking things up off the ground, as opposed to bulk sampling. There's huge biases in terms of geography and where fossils have been found. And very, very importantly, the inability of researchers to identify things that are not complete. Generally, this requires the experts in the field to look at them, and generally speaking, those are few and far between. You might think of it like this. Here's a modern ecosystem outside of St. Petersburg, Russia. So I would say, okay, what's the biodiversity here? Well, there's several ways you can look at it. Let's just simplify things and say animals. Well, what do you mean? Do you mean the animals that live in the trees? The animals that live in the mudflat? The animals that live in the water? Those are different things. And maybe your research methods will pick one of them. Or maybe it's something like, are you talking about the animals that are diurnal animals, ones that are active in the daytime versus ones that are active at night? Common animals, rare animals. The way you sample this can fundamentally affect how you perceive that ecosystem. And that is also very true in the fossil record, because the same sorts of things happen. Not these exact things, but it's a lot of decision making on what you collect, where you collect, how you collect will change the way you perceive that biodiversity. Sampling bias also shows up in geography. In 2008, this is the exact sum total of all Edreo-asteroid fossils from the continent of Africa. The 5 fragments on that rock. And then in 2008, somebody described something like that. And then Samuel Zamora went to Morocco. And in the course of a couple of weeks, he found this one, this one, this one, this one, this one. This could go on for hours. He found all those species of edrioasteroids in an area about the size of Knoxville. Why is that? Because no one had looked. So now the edrioasteroid fauna of Africa is as diverse as the fauna of the Ordovician of the mid-continent US. But we didn't know that until it was actually sampled. And the environments that they occur in are fundamentally different. Edrioasteroids prior to this work were assumed to be tropical, living in carbonate environments. These were sitting on the South Pole, 89 degrees south in siliciclastics. Very different environment. So that sampling bias fundamentally affected the way we see things. We also have biases that are associated with the fact that we're dealing with fossils. Now, one thing that's true about fossils is if you have a tree of life, and this is just a diagram of an evolutionary tree, and time goes up. If you sample this at different time intervals and you have good sampling, you will see that there's a pattern of diversity. At this timeline, the lowest one, time 1, we have 3 fossils. Here we have 8. The next one we have 14. The next one we have 11. The next one we have 6. So the diversity expanded and then contracted. If you don't have a fossil record, you get a fundamentally different pattern every single time. If you go top down using the tools of biology like genetic data and things like that, you will only see an inverted cone of diversity because the lineages that are highlighted in red are visible from a top-down view, and the ones that are highlighted in yellow are invisible to that toolbox. And so, whereas here in yellow, over here we have expansion and contraction of biodiversity, here we have increasing biodiversity. So that fundamentally changes things. And this is actually one of the main reasons a fossil record is important to study. So with all of that, we have to address a few questions. Is this pattern robust enough that the way we sample is not influencing the way this pattern is perceived? So I'm going to talk to you about a group of organisms that has a bit of a checkered history in terms of its preservation. If you think about a thing like a snail, this is a snail. If it dies, how many parts are left? 1, right? Simple enough. And if you saw a snail shell, you would say, oh, a snail. It's not rocket science. This is an echinoderm. It's a starfish. I would argue that this animal has a few more than 1 part. A few different counts you do. Most echinoderms have between 10,000 and a couple of million parts. And most of them are ridiculously tiny. When they die, this is what they turn into. This is a starfish soaked overnight in bleach. And the human mind is very good at simplifying, and the way you simplify this is you call this sand. And when you call it sand, it goes away. And if you looked at this, there are only a handful of people on Earth that could make any sense of this as anything other than echinoderm hash. If you want to know more about this, I've actually made a couple of videos on my YouTube channel about this. So just drop by and you can see, those are the 2 videos that relate to this. How many plates does an echinoderm have, where I have a time-lapse video of an echinoderm exploding, which is fun. And just another video there. Anyway. So this is what echinoderms almost always look like in the fossil record. This is called an encrinite. The name comes from the fact that it is a whole bunch of crinoids. And I think everybody knows what a crinoid looks like. You know, poker chips with a hole in it, right? But even a cursory glance, I'm hoping that you can see that, you know, this crinoid has a circular hole. This one has a pentagonal or star-shaped hole. This one has a pinhole. This one, the hole has a lip on it. There's a lot of different kinds of crinoids in here. So if you just say crinoid, it makes it sound like your diversity is one species of crinoid, when really it's a lot. If you look harder, you can see things like this. This is a diploparieton. These were last looked at in our department by Sarah Sheffield. And again, it's something that not a lot of people would jump out at. And over here, we have plates of a rhombiferan. There's also blastoid plates in here. And if we put this under a microscope, I'm sure we could find a lot more things. But you really have to know what you're looking at in order to make any sense of this. So it's not a simple process. So what I'm going to argue is you can actually identify these elements if you know what you're doing. And the way you do this is actually fairly simple. If it's an organism that's alive today, you can simply disassociate one, and then you understand what the parts are because you have them loose and free and you can look at them at your leisure. If they're an extinct group, it's a little more complicated. You have to use some very careful comparative anatomy. You look for specimens that are well preserved, except for an area that are disrupted, where you get things jumbled, so you see different angles at things. You look for elements that are serially repeated. Serially repeated elements are common, and so you're more likely to find them in a pile of hash. You look for elements that have something very distinctive about them. And what's left, you work out with a process of elimination while building a body of knowledge and experience. I can tell you from my personal experiences working with students that there's a pretty steep learning curve, but once you get there, it's really amazing how quickly you can actually identify different types of parts just through having seen a bunch of them. So this is a brittle star, and this is just an example of how we look at something. Obviously, this thing has, I don't know, 10,000 parts, let's say. So how do you deal with it? Well, if you go in and look at the mouth, what you see right off is there's lots of the plates. In fact, most of the plates are these little tiny platelets over here. There's no way you'd ever be able to know what those are. They're just small, nondescript little plates that are, you know, 50 microns across. There's plates in the mouth area that are very distinctive. These plates right here, for example, are very distinctive. There's 10 of them on the whole animal. And so you're dealing with 10 plates out of, let's say, 10,000. So you're going to have a hard time finding them. If you go to the top side of the animal, you know, you don't have a lot of help there. You just got a whole bunch of stuff. But now let's look at the arm itself. The arm is a whole series of serially repeated segments. Because of that, we have a series of plates here like these. These are called dorsal shield elements, and they repeat right and left, but they're orderly and understandable. We have these plates here with the spine bases on them, here, here, and here, and that's called the lateral arm plate. Again, those plates serially repeat, so we have a lot of them, and because of that, they're orderly and understandable. They're also very distinctive. The bottom, here's our ventral shield plates. A lot of things that look the same, but again, common. And the spines, again, orderly, but not distinctive in any way. So the bottom line is when you look for parts of an animal like this, research has shown in the Mesozoic that The primary plate you're looking for are the lateral arm plates where the spines attach, and the plates that core the center of the arm called the vertebrae. Those plates are distinctive and we can understand them. They look like this. This is a vertebra element. All these holes, that's the microstructure of them. That's what they look like when they're, because those are full of the soft tissues. This is one of the lateral arm plates. Look at all the morphology here. You have these, you can count the number of spine bases. The spine bases have some interesting sculpture, different pores that things come in and out of, different types of microstructure where the plates overlap each other or form the body wall. So there's a lot going on here that you can learn from. And because we can understand these in the modern, we can find the same things in the past. This is a Pennsylvanian form. Here are the spine bases right here with all those interesting morphologies. On the inside, we see all the little articulation things. And look at the preservation of the microstructure. I mean, these are beautiful. This is the kind of thing you just die to see. Okay, so I've put up the straw man that isolated plates can change our perception of biodiversity. So what we're going to do now is we're going to go to a single locality and see how that single locality, based on how you sample it, will fundamentally change our perceptions of biodiversity at different scales. This locality is in the Glen Dean Formation, which is upper Mississippian. That locality, or the Glen Dean Formation, has been studied from countless localities all over the state of Kentucky. And a summary from Chestnut and Ettinson and Chestnut suggests that they have 37 species of crinoids, 10 species of blastoids, 1 species of edrioasteroid, 2 sea urchins, 2 brittle stars, and a starfish. These are all from whole specimen data. So people going out, finding a slab of rock with one of these animals stuck to it. We're going to go to a little road cut right here north of Hopkinsville, Kentucky. So here's Nashville. This is up here. Okay. To say that this locality is full of blastoids is an understatement. In this picture, there's 3 blastoids just sitting on the ground. You can go out there with 5 of your closest friends and leave with 2,000 blastoids in an hour. It's that dense. I've been there maybe 5 times with students on all these occasions. We probably spent about 30 man hours going over this road cut. And this is the echinoderm fauna we found in this road cut. 2 species of crinoids, 5 species of blastoids, including one that Chestnut and Edison didn't have. Nothing else. So from this perspective, we can say, wow, this locality's got an awful lot of crinoids in it, or blastoids in it, and not much else. Here's the road cut. This shale unit is where all the fossils are coming from. This is an oolitic shoal up here. Beautiful, beautiful grainstone. And you just walk around and you pick up things, you move the car doors around as need be, and collect your fossils away. And like I said, it's just amazing how many fossils are here. Now, we surface collected, but we also brought back bulk samples. You wash out the bulk samples, and this is what you get when you wash all of the clay out. So all of this, every bit of it is a fossil grain. Crinoid here, blastoid part here. Here's a whole blastoid, bryozoans, whole 9 yards. So rich, diverse fauna. The problem is you can't look through that because it drives you crazy, because the big things make it hard to look for little things. So you have to sieve it out. And when you sieve it, it looks like that. And then you simply go through that material under the microscope, picking material by hand. And you wind up with little slides covered with very exciting, if you care, echinoderm parts, okay? So it's a long process to get here, but you wind up with an awful lot of material at the end of the day. So the crinoids at this locality are the most common echinoderm element. I am not a crinoid worker, so I cannot say anything terribly useful about the crinoids. From whole specimens, we only found 2 species, but there are lots and lots of different kinds of plates at this locality that I couldn't actually identify. Mississippian crinoids at this time are among the most challenging crinoids when you have whole ones, and having isolated fragments is really a specialist job, not mine. For those of you who don't know what a crinoid is, here's a modern crinoid in the Great Barrier Reef. It was so cute sitting there. Aw. But we see that. And again, you know, even not knowing a lot about crinoids, I'm sure you can see that this stem is very, very different from this stem. This stem is very different. This stem is very different. So, and then these are plates of the body of the calyx, all these little bits here, arm elements. We got the whole animal. It's just a mess. And I'm not gonna, I can't put a number on this. I don't have that skill set. But from what I see here, there's certainly lots of things. What we can add to the fauna are microcrinoids. This is a group of crinoids that are only 1 millimeter high when they're mature. And we see lots and lots of these. So this is something, again, you wouldn't see just crawling around in your hands in these. Although I had a student once pick one of these up in Texas. Seriously, they just went poop and picked up a fossil that was 1 millimeter across off the outcrop. Blastoids. Again, blastoids are extremely abundant here, literally thousands of them. But the nice thing is we understand the morphology of a blastoid, so it's relatively easy to pick them up. What's also present in huge numbers are larval blastoids, what's called Paleozoic. It's not so much a taxon as much as an age category. This is the most common blastoid there, the Pentramites, and you see the beautiful preservation at this locality. But based on the morphologies you can see here, you can identify all the different parts of the blastoid from the sediment. This is a basal plate. These are lancet plates, deltoid plate, okay? Pretty straightforward stuff. This is the Paleozoic. You can see the scale on this, that's 200 microns. That's the top of the blastoid, and there's 3 stem columnals coming down off the bottom of it. This is actually the most common echinoderm at the entire locality, believe it or not. I had a student do undergraduate research, and from a couple of samples weighed and washed, calculated 37,000 of these per kilogram of sediment. So these are amazingly abundant. But again, 37,000 things that are 200 microns high isn't actually all that much material. Sea urchins. We didn't see any sea urchins at the locality, but we have lots of sea urchin parts. We have them from all over the animal, the tests, the spines, the lantern elements. They're all there in the sediment if we know what we're looking for. So that's a modern sea urchin. You can see on the outside we have some of these pieces with 2 holes. And then we have these ones with the big knobs on them. These are where the spines attach. And then here's material from the sediment. There's one of the plates with 2 holes in it. Here's a spine. And there's one of the ones with the knobs on it. So we have the whole animal from the outside. This is the inside of a sea urchin. That's the lantern, that's the mouth apparatus. And you can see it has a big tooth here. This is called a pyramid. And it has this called a rotula over here and some other elements. And in the sediment, we have these. That's the pyramid, that's the rotula, that's the tooth, that's one of the upper elements on the lantern. So all the parts are there, but you wouldn't recognize them unless you knew what you were looking for. Holothurians are sea cucumbers. Sea cucumbers are echinoderms, and they have huge number of skeletal elements embedded in their body wall, but they're exceedingly tiny. That's a modern sea cucumber. Doesn't kind of look like a crinoid very much, but these are sea cucumber parts. This is a wheel element, this is an anchor element, and this is one of the 10 plates around the throat of the animal. Again, you see the scale on these things. These are very, very small, but they're in the sediment along with everything else, if you know what you're looking for. I cut my teeth on edrioasteroids. Edrioasteroids are the first organisms I worked on trying to straighten out what the individual elements look like. While we have no edrioasteroid specimens from this locality, we have hundreds and hundreds of plates, and they're very easy to recognize once you know what they look like. These are whole specimens of the two types we have. This is called a discocystenid, yeah, disco. And this is a lepidodiscenid. These are some of the plates from the arms. These are what moves the food along, that covers it. This is the edge of the mouth frame. These are plates from the bottom of the animal. This is what's called a recumbent zone plate. That's part of the holdfast, and this is the flexible stalk. The plating of the body wall, and this is the plates that cover the anal opening. Not many on these on an animal, and there you go. You've now seen an anal plate of an edrioasteroid. Your life is complete. The other species, I only know it from a few elements of the arms. I know what this is, and I know it doesn't look like much to you. There's really nice dolomite here, but based on this imbricated edge here, I can tell you it's Lepidodiscus. So I can identify this one down to the genus pretty easily. So those organisms all had fairly, I would say, not unusual levels of diversity. What I didn't expect when we started this project, and in fact, when you go to Nick Smith's defense on Wednesday, you'll see a lot more of this from another locality where Nick is working, is you'll see that the brittle stars, the ophiuroids, have a diversity that is unmatched in the Paleozoic, but we only know about it from these disarticulated elements. At this particular locality, we have 11 taxa, but we don't have any articulated specimens. The lateral arm plates in the vertebrae are ubiquitous in this material. They're very easy to identify once you know what they look like. And very importantly, if you look at semi-contemporaneous localities, which we have, what we're seeing in this locality is not abnormal. This is typical. Nick has the same level of diversity in the Sulphur, Indiana locality. We have nearly the same diversity in a locality right outside of St. Louis that's about the same age. So this is normal diversity for this. And that would be a brittle star. We have a couple of different species at this locality of things called stenuroids. Stenuroids, lateral arm plates sort of look like rackets with a peg on the end. And these are the little facets for the spines coming off. Interestingly enough, this is Upper Mississippian. The youngest known stenuroids in the literature right now are in the Devonian. So this extends these from the end of the Devonian all the way through the end of the Mississippian. And we have diversity. This is the most common brittle star at this locality, it's this group. We have a protasterid. which is kind of a weird looking thing with this big stereome here, and these are the spine facets. Another protasterid, which varies clinically in where the spines attach and some details here and there, but it's a different one. We have a thing called cloaster. This one's the big one. So you can see that these plates here are pushing 2 millimeters across. So this is the only brittle star where the plates are over 0.5 millimeter across. These are the vertebrae of the same. We have 2 species of furcasterid. This is a sort of a sickle-shaped guy here. We call it species A because why not? We have the vertebrae that we can associate with this based on a couple of specimens that were given to us by a collector in Alabama. This is the other furcasterid we have here. It's a lot longer plate. It's not so sickle-shaped. And we have the associated vertebrae here, again, with that really nice preservation. We have an aganaster. An aganaster belongs to the modern group of brittle stars. And the vertebrae associated with that appear to be this thing, which frankly, I think looks a lot like a giraffe skull, but that might just be me. We have this new thing that Nick's naming called sulfaster, named after Sulphur, Indiana. And again, this one's exceedingly small. This is just 1/4 millimeter long. And we have a couple things that we know they're brittle stars based on their morphology, but they're really defying our ability to classify them right now. And we just put it in Incertacetus, which is Latin for I don't know what it is. But it's a different kind of brittle star that we haven't seen. This is another weird one. It has these 2 great big facets on the front, and we're not really sure what this attaches to, but it is different from the other ones that we've seen. And it's often very difficult to place the vertebrae with the lateral arm plates. So we have a lot of vertebrae that we don't know what they belong to. You can identify the taxonomies based on the lateral arm plates. So we just have these. We call this one the plucked chicken. Kind of looks like a roasted chicken. So what we see then, I hopefully have shown you, is that there's a fundamental difference in the level of diversity we see at this locality depending on the methods we use. If we go back to our original 30 man-hours crawling over the outcrop level of trying to see what the echinoderm diversity is, we had 2 species of crinoids, we have 5 species of blastoids, and nothing else at this world-famous fossil locality. This is what we add if we add in what we know from isolated elements. Ignore what the crinoids are. The crinoids would probably be way off the graph here, but I don't have the skill set to understand that crinoid data. The blastoids didn't change, but we added echinoids, 2 species, edrioasteroids, 2 species, sea cucumbers, 2 species, and 11 species of brittle stars. That's fundamentally different. So at least at a very initial level, we can say that yes, if you only sample haphazardly on the surface, you will get a diversity pattern that is muted with respect to what is actually there if you look and you know what you're looking at. But can this change the global pattern? Well, I think it can. Here's the fauna from the Glendine. Over here, we had 37 species of crinoids described by Ettinson and Chestnut. we added 1. We probably could add more if I knew anything about crinoids. Blastoid fauna, we haven't really changed it that much. We added 2 new edrioasteroids to the fauna. We already had the 2 echinoids. We added 2 sea cucumbers and 9 species of brittle stars to the fauna. So this formations diversity has skyrocketed in terms of the ophiuroids, which went from relative obscurity to the second most abundant group of echinoderms because we looked at it in a different way. And then if you bump this up even further, this is the famous echinoderm compilation of diversity from Sprinkle. And these spindle diagrams are, just think of these as bar graphs. So this shows you the relative amount of diversity at different times through Earth's history by the width of the bar, okay? And the pattern of ophiuroids is actually very interesting. It's really low level diversity throughout the Paleozoic until you get to the end of the Mississippian. And then it's basically nothing. There's a couple taxa here until you get well into the Triassic. What happens when we look at it? The highest level of diversity in the Paleozoic is that one locality. There's more diversity in the Paleozoic than any other time in the Paleozoic when you sum up every brittle star that's been described everywhere in the world. It's staggering the changes you see here. And then you put it up with against the diagrams like this. These are the plates that are going into the proposal I'm working on of just all the different types of diversity here. And then, if that weren't enough, Nick has 11 species in his locality, and those 11 species include 3 I don't have. And they're the same age. So really this bar is even wider if we put all the data in. So what we're going to do from here on out is we're going to try to duplicate this throughout this section and see what the real diversity is in brittle stars through time. And what's going to happen based on a few initial samplings that we've done is it's all going to look like this, all up here. So we're fundamentally rewriting the diversity of a group through time because we're using a different methodology. So in conclusion, echinoderms that have very flexible skeletons with huge numbers of body parts are going to always be underrepresented in the fossil record because there's a specimen bias where we study specimens that are complete rather than studying specimens that have disintegrated. Although these skeletons can fall apart, some of the elements are diagnostic and can be identified at the species level. Taxon counts based on disarticulated faunal elements can affect local diversity and the estimates for global diversity compilations. In other words, how we collect data from the fossil record really does matter. Okay? Oop. And I'll leave it there. And I'll entertain questions for a while.
Hey, Colin. This is Steve Romanello.
Hey.
I have a question.
Yep.
So totally sold on this idea, right? And I think this is awesome, and I think you're totally right about it. But the question is how to implement it practically, right? Because we've got so much rock to look at, and it takes such specialty knowledge for each group of organism, and you really have to do a lot of picking and separating, right?
Yep.
So I'm curious what you think of efforts to use automated image classification systems, like the way that Google finds pictures of cats, right? Can we train, if there was a substantial effort, let's say I don't know how many $1 million effort, could we train automated detection systems to look at these microfossils and begin bit by bit to sort of, because a human will never be able to look at all of these. A human will never be able to aggregate everything we know about the fossil record now, but a substantially scaled computer algorithm probably could, right?
I would say the answer is yes and no. Yes is once we understand what we're seeing, yes. But the way the brittle star portion of this project evolved, and you can talk to Nick about this, he was much more involved in the brittle star part, was when Nick came in, I said, I've got this project. We're going to look at brittle stars. And he said, and I said, there's only two problems. One is no one has actually done any work on this, so we don't know what anything you're looking for actually looks like. And the second part was, I don't remember the second. Ask Nick, he'll remember it. And when we started doing this, we were working with a colleague in Luxembourg who'd only worked in the Mesozoic. And so what kept happening, he said, you'll know them when you see them. So we were just going back and forth between the SEM and emailing pictures and saying, is this one? Yes. Is this one? No. Is this one? Yes. And we thought we had it all worked out. We had 6 or 7 species. And then Nick went to Luxembourg and handed him some sediment. He put it on a tray and says, well, you missed this one and this one and this one and this one. So we are still trying to understand what these things look like. Once we know what they look like, they are distinctive. So when every time we find a new type and we get it imprinted in our brains, we can see it. And from that aspect, I think you're right, there could be a way to do that. But you can't do that until you actually know what they look like.
Yeah, I think the problem is that everybody's got a couple dozen of them imprinted in their brain, and we need them all imprinted in one place.
Well, everybody is me and Nick.
Yeah. So, interesting.
So a lot of what you were discussing was size bias. So what do you find when you're crawling on the outcrop versus what do you find when you actually are sieving it?
Yep.
Coming from my dinosaur project, how much of an issue do you have in echinoderm paleo with basically trophy hunting collection? So only bringing back the most beautiful specimens and leaving behind even identifiable stuff in the field.
That's huge, yeah. In echinoderms, there's a big problem with trophy hunting for 2 reasons. One is trophy hunters take good stuff, and the second problem is that's what gets into the museums. As a result, when you're looking at a brittle star that's perfectly preserved, you can't actually see a lot of the morphology that you would need to see, like the inside of the plates, like how the plates articulate on those articulation surfaces. We need the mangled up specimens to be able to see the important things. And so when you only have the really good stuff, you actually can't see. It's sort of like what your advisor told me once upon a time about this croc was so well preserved I couldn't see anything because all the osteoderms were covering all the important parts. And that's a huge problem. I mean, I've always said with edrioasteroids, give me one really nice, beautiful specimen and 12 that are mangled in various exciting ways, and I'm a happy camper.
Colin?
Yeah.
These are very small. I wonder how you do with potentially reworking of sediments or distribution of these particles, because they might be from very different places than they end up being deposited.
There's actually been a fair bit of work on crinoid disarticulation in the Great Barrier Reef. And what we find is that most crinoids, for example, are incorporated into sediment in situ, which I don't think, I find that a little surprising, but in terms of reworking, they're certainly not being reworked up section all that much. There's of course going to be some local time averaging, but it's probably not going to be very extensive. These are also being deposited in a clay-rich shale. I mean, so the depositional environment is very, very, very low energy, at least locally here.
Hey, Colin, similar question. Have you done any work, because these are so small and you're getting such a big sample from just one little pile of sediment, have you done any work to see if you like sample on one side of the outcrop versus the other, if you get different answers?
We've done that, and you get the same answer in we brought back like 20 bags of this stuff, and they're all the same. All the same, within air bars.
Colin, I'm thinking about the imaging problem a little bit more so all this is based on visual imagery, but if you mapped it chemically, for example, or you went in with other techniques, hyperspectral techniques or something like that, do you think there'd be additional information you could glean at looking at a big pile of this stuff? Would the elements stand out in particular ways, or would different species or parts match up in a way that lets you?
One would think that all the echinoderm parts might group together, but you also have to concern yourself with, crinoids have, more parts than brittle stars, and they are more numerous in the localities than brittle stars and have pieces that are of the same basic size fraction. So you're going to get a lot of swamped signals there. I would imagine that if you fingerprint them chemically, I'm sure the echinoderms would be distinctive in that they start out as magnesium calcite, and the only other magnesium calcites in there would be maybe the bryozoans and the brachiopods, which are pretty common in there. But there's probably ways to do it. But again, you still got to figure out what you're seeing, and that's the biggest problem. Okay, well, I guess that's it. And thank you all for coming. Thanks for watching, and we'll see you next time.